So this will be the last video specifically from chapter 16. We're just going to finish up with the enzyme-coupled receptors, in particular receptor tyrosine kinases. I'm going to give you an overview of activation of receptor tyrosine kinases here, but then we will also be focusing on them in the case study on insulin signaling. So insulin binds to receptor tyrosine kinase type receptors. So the case study will be looking at these mechanisms of cell signaling in more detail. Previously, I introduced you to enzyme-coupled receptors more generally, and in the case of some of those receptors, the signal molecule was a dimer. And that's the case here with the receptor tyrosine kinase. And binding of the dimer initiates kinase activity in the receptor itself, in this homodimer. Remember, both parts were the same. Each half of the receptor is then activated as a kinase, which can phosphorylate the other on particular amino acids that are tyrosine residues. This phosphorylation creates a new shape of the receptor in the intracellular surface. So prior to being activated and phosphorylated, the shape of these receptors were different in the intracellular surface. Once we have created this new shape, or once binding of the signal creates the new shape, proteins can then bind to the receptors. So it kind of creates what is called a scaffold for binding of signaling proteins. Here they're showing 1, 2, and 3. And here's a specific pathway that's being activated. We have phosphorylation of the receptor, binding of an adapter protein, which activates a GEF, a guanine nucleotide exchange factor. This guanine nucleotide exchange factor is acting upon a small G protein called RAS. RAS is very similar to the alpha subunit of the trimeric G protein if it were just by itself, and it has a lipid link attaching it to the cell membrane. Like any other G protein, it's inactive when bound to GDP. The RAS GEF promotes its release of GDP and the exchange for GTP, which activates RAS, which then sends the message onward further in the pathway. It happens to be that RAS can activate a kinase cascade, meaning one kinase phosphorylating and turning on another kinase and another kinase. So we may actually want to go to, this was probably the first one discovered. It's called MAP kinase, mitogen activated protein kinase. Mitogens are things that promote cell division, for example. And RAS is known to be involved in cell division. It's something you likely discuss with Dr. Smith when you're covering cancer and the cell cycle. So you can see that this MAP kinase is phosphorylated by a protein called MAP kinase kinase, and MAP kinase kinase is phosphorylated by a protein called MAP kinase kinase kinase. So this is our cascade of phosphorylation. RAS activates MAP kinase kinase kinase, which activates MAP kinase kinase, which activates MAP kinase, and then MAP kinase can phosphorylate a number of different target proteins that may cause fast effects, things that happen fairly quickly, like changes in protein activity, and also effects that take longer, like transcription and translation. We will see a modified version of this receptor tyrosine kinase pathway in the insulin signaling case study that we're going to do on Monday. Just one other note about cell signaling is that there are signals that do not use membrane-bound receptors. Things like these hormones, cortisol, estradiol, testosterone, thyroxine, these are all small hydrophobic molecules that can cross the phospholipid bilayer. Because of that, we're looking at cortisol as an example. The receptor may be within the cytoplasm, and in the case of others, sometimes in the nucleus itself. We can see here for cortisol, cortisol meets up with its receptor in the cytoplasm and then translocates into the nucleus, where the receptor itself is a transcriptional regulator and can turn on gene expression. So while a lot of our cell signaling pathways are occurring at the cell membrane, some very important cell signaling pathways are occurring within the cell. The receptor binding to the ligand is happening within the cell, and this is often the case when we have the receptor being itself a transcriptional regulator, turning on or off gene transcription. And I'm throwing in this last slide because I do think it's important to note that multiple signaling pathways are happening within your cells at one time. We're not just having cortisol or adrenaline or insulin happening to one cell at one time. Cells are interpreting what to do based on the cumulative signals they are receiving at one particular time. And you can see some of these signals can actually be contradictory. Signal D is promoting the activity of kinase 2, whereas signal C is inhibiting it. But we also have signal B promoting it, right? So there's a titration effect where how much signal B, how much signal D we have promoting the activity of the kinase, does that overcome signal C? If we turn off signal C, we probably get a stronger response of the cell. We can see that kinase A also has two pathways activating it, and that these all come together in one target protein to have a cell response. So the complications, the interactions between these cell signaling pathways means that the cell can have a very precise response to particular signals, sometimes a stronger response, sometimes a less strong response, something that's the response should be proportional to the things that are happening in its environment. So that's going to be the end of chapter 16 as far as the textbook resources, but we will be, again, looking at the case study on insulin signaling and the relationship to diabetes. That is a two-part case study, and in the first part it does review general properties of cell signaling that were already introduced here, and then in the second part focuses in on receptor tyrosine kinase signaling and the insulin receptor. I will likely post a little bit of an introduction to that case study. There are a couple of figures I want to make sure that you are familiar with and that the pictures make sense when you're working on the case study.